interpretation of the results. During the PCR, the product size should be of expected size. Misprime may occur due to the attachment of the primers other than the original point of the attachment because they are oligonucleotides. If they do not attach to the original location and they attach to other locations of the DNA, so as a result of this mispriming, other regions on the DNA can also be amplified. As well as primers, they are oligonucleotides. One is the left primer and the second one is a right primer. So primers, they can join with each other, and there can also be primer dimers. So while performing a PCR reaction, for the better interpretation of the results, there should be a blank reaction. What is a blank reaction? Blank reaction is actually to control the contamination that contain all the reagents except the template DNA. One is the main reaction in which there are all reagents as well as template DNA, but in this tube that is called as blank reaction, there should be all the reagents. There will not be DNA in this reaction. So that reaction is called as blank reaction. In addition to blank reaction, there should also be negative control. Negative control is for the specificity of the amplification reaction that contain all the reagents, a DNA template lacking the target sequence. So in this reaction, there are all the reagents, there is a DNA, but that DNA lacks the target. So that type is called as negative control. Here is the diagram. We will try to understand as a result of PCR reaction. We can see that this is the blank reaction. Here we can see that nothing has been amplified. So here there is empty. So the next lane is for the positive control. Here we can see this is the positive control, and in this well there is amplification of the DNA. This one was the negative control. Because this is negative control, so there should not be any kind of amplification in this well. So this is the result of patient 1, patient 2 and patient 3, these 3 patients. And this is the DNA ladder, or also called as molecular marker. So the expected size of the patients for a particular region of the DNA was around about 1000 base pair. So if we see that whether our expected band is of the same that what we were expecting as a result of PCR reaction, if we see that this band of the molecular marker, this is 100, 200, 300, 400, 5, 6, 7, 8, 9, and this is the band of 1000 base pair. So our expected size of the band of the patients was approximately 1000 base pair. So we can say that this is the actual region has been amplified. So while performing the PCR reaction, there should be a blank reaction, there should be a positive control, a negative control, as well as a molecular marker should be used to interpret the results or to see whether the expected band size is of a required base pair. So here we can see the results are according to our expectations. So if we conclude that the PCR product as a result of PCR reaction, the expected size of the band should be of same size what we were expecting before performing the PCR reaction.